Hello dear students, welcome to the botany online class. So this is the continuation of previous video. In that we have studied about the structure of megasporangium or structure of ovule, types of ovules and placentation, right? Okay. So today we are going to learn about the new topic, that is the process of megasporogenesis and formation of female gametophyte. So what do you mean by megasporogenesis? To explain this topic, let me recall the points of structure of megasporangium. Okay, so as I said before, megasporangium, it is also called as ovule, right? Now, each ovule, it contains several parts. The stalk of the ovule is called as funicle. Now, along with the funicle, the entire ovule, it consists of two protective layers called integuments. So, integuments are nothing but it is the protective layer of the ovule. Okay. Now, at the tip, the ovule contains an opening, and this opening is called as the micropyle, and just opposite to the micropyle there is a chalaza, and this chalaza is the basal part of the ovule. Am I clear? Now, after the integuments, the ovule contains a nutritive portion, and this nutritive portion is called as the nucellus. So what is the function of the nucellus? It gives nutrition. Okay. Now, in the nucellus, it contains a female gametophyte, and this female gametophyte is also called as the embryo sac, and within the embryo sac, it consists of three groups of cells. At the micropylar end, it consists of egg apparatus. At the chalazal end, antipodals are present, and at the center, secondary nucleus is present. So these are about the structure of megasporangium, right? Okay, coming to the process of megasporogenesis, what happens is, in young ovule, there is no embryo sac within the nucellus. That means within the nucellus, embryo sac is entirely absent, or female gametophyte is entirely absent. So during the developmental stages, any cell of the nucellus at the micropylar region, once again I will repeat, during the developmental stages, any cell of the nucellus at the micropylar region enlarges in size and it functions as the megaspore mother cell. Am I clear? So this megaspore mother cell later on undergoes division to form megaspores. Am I clear? Any doubts? So once again I will repeat, during the developmental stages, any cell of the nucellus at the micropylar region enlarges in size and it becomes megaspore mother cell. Now this megaspore mother cell is diploid, correct? Okay. Now what happens next? This megaspore mother cell, it undergoes division so that 4 megaspores are formed. Am I clear? So from a single megaspore mother cell, 4 megaspores are formed, and all these 4 megaspores are haploid in nature. Clear? Now, what happens next? So this is the process. Here, this is the single megaspore mother cell, which forms from the nucellar cell, and this megaspore mother cell, it undergoes division so that finally 4 megaspores are formed, and the process of formation of megaspores from megaspore mother cell is called as megasporogenesis. Once again I will repeat, the process of formation of megaspores from megaspore mother cell through meiosis is called as Megasporogenesis. Am I clear? Okay, at the end of Megasporogenesis, how many megaspores are formed? 4 megaspores are formed. Is that clear? Okay, next, of these 4 megaspores, 3 megaspores degenerate and only 1 megaspore becomes functional. Once again, I'll repeat. At the end of Megasporogenesis, from a single megaspore mother cell, 4 megaspores are formed. Clear? Out of these 4 megaspores, only 1 megaspore becomes functional and the remaining 3 megaspores become degenerate. Is that clear? Okay. Now what happens next? In majority of the flowering plants, 1 megaspore is functional while the other 3 degenerate. The functional megaspore develops into the female gametophyte. Okay, so the functional megaspore, it develops into the female gametophyte, and the embryo sac or the female gametophyte formation from a single megaspore is called as monosporic development. So what do you mean by monosporic development of embryo sac? If an embryo sac is formed from a single megaspore, then it is called as monosporic type of embryo sac development. Is that clear? Okay. Now what happens next? So here you can observe the process from a single megaspore mother cell through meiosis. Megaspores are formed. Till here it is called as Megasporogenesis. Now after the formation of 4 megaspores, the 3 megaspores degenerate, whereas only 1 megaspore becomes functional. Okay. Now this functional megaspore, it contains haploid nucleus. Okay. Now what happens? The functional megaspore, it undergoes nuclear division. As a result of this, 2 nucleate, 4 nucleate, 8 nucleate embryo sac is formed. Okay. Initially from a single nucleus, 2 nuclei are formed. Later 4 nuclei are formed, and finally 8 nuclei are produced. Now out of these 8 nuclei, 3 nuclei move towards the chalazal end and forms the antipodals. Once again, I'll repeat. Out of 8 nuclei, 3 nuclei move towards the chalazal end and forms the antipodals. Then again 3 nuclei move towards the micropylar end and forms the egg apparatus. In the egg apparatus, it consists of 1 egg cell and 2 synergids. And finally, the remaining 2 nuclei at the center forms the secondary nucleus. So in this way, the embryo sac is formed within the nucellus. And the process of formation of embryo sac from a single megaspore is called as monosporic type of embryo sac development. Is that clear? The nucleus of the functional megaspore divides mitotically to form 2 nuclei. They move to the opposite pole, forming 2 nucleate embryo sac. Later, the nuclei again divide 2 times, forming 4 nucleate and 8 nucleate stages of the embryo sac. These divisions are free nuclear. That means nuclear divisions are not followed immediately by the cell wall formation. After the 8 nucleate stage, the cell walls are laid down, leading to the organization of the typical female gametophyte. 6 of the 8 nuclei are surrounded by the cell walls and are organized into cells. And the remaining 2 nuclei are situated at the center, form the secondary nucleus. And a typical mature embryo sac is 8 nucleate and 7 celled. Is that clear? 3 cells are grouped at the micropylar end, form the egg apparatus. And this egg apparatus consists of 2 synergids and 1 egg cell. Now the synergids have special cellular thickenings at the micropylar tip called filiform apparatus. Once again I will repeat. The synergids have special cellular thickenings at the micropylar tip, and this special thickening is called as the filiform apparatus. Now what is the function of filiform apparatus? It helps to guide the pollen tubes into the synergids. The 3 cells at the chalazal end forms the antipodals, and the large central cell called the secondary nucleus has polar nuclei. So this is the structure of mature embryo sac. So once again I will recall the points. At the time of development, there is no embryo sac within the nucellus. But later what happens is, any cell of the nucellus at the micropylar region enlarges in size and forms the megaspore mother cell. And this megaspore mother cell is diploid. Okay. Now, the megaspore mother cell undergoes meiosis. As a result of this, 4 megaspores are formed. And this process of formation of megaspores from megaspore mother cell through meiosis is called as Megasporogenesis. Am I clear? Okay. Now what happens next? After the formation of 4 megaspores, only 1 megaspore becomes functional and the remaining 3 become degenerate. Okay. Now this functional megaspore, the nucleus of the functional megaspore, it divides repeatedly to form 2, 4 and 8 nuclei. Now later on, of these 8 nuclei, 3 nuclei move towards the antipodal end, the chalazal end and forms the antipodals. Again 3 nuclei move towards the micropylar end and form the egg apparatus, where it consists of single egg cell and 2 synergids, and the remaining 2 nuclei at the center forms the secondary nucleus. Am I clear? And a typical embryo sac, it is 7 celled and 8 nucleate. Okay, and this process of development of embryo sac is also called as monosporic or polygonum type of embryo sac development. Am I clear? So this is the picture of process of megasporogenesis and the formation of female gametophyte. So here you can observe the process. So this is a single megaspore mother cell which divides to form megaspores, 4 megaspores. So of these 4 megaspores, only 1 megaspore becomes functional, and the nucleus of that functional megaspore, it divides repeatedly to form 2 nuclei, 4 nuclei and later on 8 nuclei. Now after the formation of 8 nuclei, within the embryo sac, 3 groups of cells are formed. They are antipodals at the chalazal end, egg apparatus at the micropylar end, and in the egg apparatus it consists of single egg cell and 2 synergids, and at the center it forms the secondary nucleus. Is that clear? So these are about the process of megasporogenesis and the formation of female gametophyte. So moving on to the next concept. Now, at the time of development of the female gametophyte, what happens is the nucellus cell or any cell of the nucellus at the micropylar region, it enlarges in size and it becomes the archisporial cell. Once again I will repeat, any cell of the nucellus at the micropylar region enlarges in size and that cell forms the archisporial cell or archisporium cell. Is that clear? Okay. Now, based on the behavior of the archisporial cell, the ovules are classified into two types. Once again I will repeat, based on the behavior of the archisporial cell, the ovules are grouped into two types. They are crassinucellate ovule and tininucellate ovule. Am I clear? Now, what do you mean by crassinucellate ovule? So, in this type of ovule, the nucellus is very well developed. That means, so as I said before, any cell of the nucellus, it enlarges and forms the archisporial cell, correct? Okay. Now, this archisporial cell, it divides to form the outer and the inner cell. The outer cell is called as the primary parietal cell and the inner cell is called as the primary sporogenous cell. Okay. Now later on, the primary parietal cell divides to form well-developed parietal tissue or nucellus and primary sporogenous cell behaves as megaspore mother cell. Okay, so once again I will repeat, in crassinucellate type of ovule, what happens is the archisporial cell, it divides to form outer primary parietal cell and inner primary sporogenous cell. Clear? Okay. Now, this primary parietal cell again divides to form parietal tissue or the nucellus, whereas the primary sporogenous cell behaves or functions as megaspore mother cell. And this type of development of ovule is called as crassinucellate. Am I clear? Okay. So here you can observe the development. So this is the archisporial cell which is formed from the nucellus. Later on, this archisporial cell, it divides to form primary parietal cell and primary sporogenous cell. The primary parietal cell again divides to form parietal tissue or the nucellus, whereas the primary sporogenous cell, it directly functions as the megaspore mother cell. Clear? Okay. We'll move on to the second type of, this is the picture of a crassinucellate type of ovule. Coming to the second type of ovule, that is tininucellate. So here nucellus is absent. That means, so at the time of development, any cell of the nucellus, it enlarges and forms the archisporium, correct? Okay. In crassinucellate type, what happens? This archisporium again it divides to form primary parietal cell and primary sporogenous cell, And again, primary parietal cell divides to form a parietal tissue or the nucellus, whereas primary sporogenous cell forms the megaspore mother cell. Correct. But in teninucellate, this archisporial cell, it does not divide, but it directly functions as megaspore mother cell. Am I clear? In crassinucellate type of ovule, there is a division of archisporium to form a parietal tissue as well as sporogenous tissue. But here there is no such division, and archisporial cell itself directly develops into the megaspore mother cell. So these two are the types of ovules based on the behavior of the archisporium cell. Is that clear? So these are about the process of megasporogenesis and the formation of female gametophyte. Now let's move on to the next topic, pollination. Pollination, it is the transfer of pollen grains from the anther to the stigma of a pistil. So it is the transfer of pollen grains from the anther to the stigma of a pistil. Based on the source of pollen, pollination is of two types. They are autogamy and allogamy. Under allogamy, again two types, geitonogamy and xenogamy. Now what do you mean by autogamy? So if the transfer of pollen grains takes place from the anther to the stigma of the same flower, once again I'll repeat, if the pollination takes place between the same flower, then it is called as autogamy. And this autogamy is also called as self-pollination. Okay, whereas allogamy means it is the transfer of pollen grains from the anther to the stigma of different flowers of the same plant or different plant. So self-pollination or autogamy, here pollination takes place in the same flower. Whereas allogamy, it requires two different flowers of same plant or different plant. Is that clear? Now under allogamy, this allogamy, it is also called as cross-pollination. And under this, there are two types, geitonogamy and xenogamy. So geitonogamy means it is the transfer of pollen grains from the anther to the stigma of another flower of the same plant. If the pollination takes place between the two different flowers of same plant, then it is called as geitonogamy. Whereas xenogamy means it is the transfer of pollen grains from the anther to the stigma of a different plant. Okay, so what is the difference between geitonogamy and xenogamy? So in geitonogamy, the pollination takes place between two different flowers of the same plant. But in xenogamy, pollination takes place between two different flowers of two different plants. Am I clear? So these are about the types of pollination, autogamy and allogamy. And under allogamy, there are geitonogamy and xenogamy. Thank you.